The diversity explored in a directed evolution experiment typically involves point mutants. By this we mean that each member of the library varies from some ancestral sequence by single base substitutions encoding single changes in amino acid identity. One common way of generating such libraries is error-prone PCR. This is just like any other PCR in terms of it containing a buffer, a thermostable polymerase, 2 oligos, and a template. The difference is that you tweak the concentrations of the DNTPs, you add manganese, and you also add additional magnesium. This set of conditions causes the polymerase to make errors at elevated rates, and it does so randomly. The conditions sometimes have the unfortunate consequence of decreasing the robustness of the PCR reaction, so sometimes you don't get any product out, or you get low yields. There are optimized commercial kits that can handle longer PCR products and do them more reliably. The GeneMorph2 kit is one that works by using a mutant error-prone polymerase. It generates a similar spectrum of mutations to error-prone PCR, but it will work on longer templates. One older method is the XL1 red strain. This E. coli strain replicates with a high error rate. Thus, you simply passage your plasmid through this strain by transformation and growing it up to introduce mutations. The rate of mutation is significantly lower than the in vitro mutagenesis methods, but the in vivo nature of the method makes some interesting experiments possible. For example, if you passage a plasmid encoding a slightly toxic protein through this strain, the selective pressure towards relieving the toxicity will result in the enrichment of mutants that solve the problem. When you sequence the variants, you will often see trivial nonsense mutations, which means stop codons, but other times you'll find point mutations or other types of errors such as insertion elements that relieve the toxicity. When you couple this with a positive selection for function, these experiments can be very informative about how to fix a toxic protein. The main issue with XL1 red is that the mutagenesis is global. The genome is also being mutated at a high rate, rendering the cells erratic and slow growers. They can be finicky to work with, and mutations to the genome can also be selected for in your experiment. A popular research topic today is trying to engineer cells that focus mutagenesis on a particular region of an introduced plasmid and thereby restrict errors to where they are useful. DNA shuffling is related to error-prone PCR. The experiment starts with a pool of point mutation variants of a gene. These mutants might be the hits identified in a previous round of directed evolution. First, you PCR up the genes with external primers. You then digest the DNA into 50 base pair fragments with DNase 1. You then recombine those fragments in a PCR reaction, and this is a very similar situation to what you're doing in PCA gene synthesis in terms of how things reassemble. You then re-amplify with external primers and subclone the PCR product into a plasmid and transform to make your library. Shuffling has largely replaced error-prone PCR in most directed evolution experiments because it does double duty. It not only recombines the mutations contained in a population of genes, it also introduces new mutations. It's error-prone itself, causing a similar spectrum of point mutations to error-prone PCR. Thus, largely, you don't do error-prone PCR anymore, you just shuffle. There are also strategies for combining computational design with directed evolution. ProSAR is one such method developed at Codexis. The first step is to generate an initial library. This can be done by error-prone PCR or shuffling. You can also explicitly synthesize each single amino acid mutant of the protein using gene synthesis. For a typical 300 amino acid protein, there are only 6,000 such variants. As microarray-based gene synthesis methods become more available, this approach will be greatly enabled, so this is probably the way it will be done in the near future. You then measure the activity of each variant. In this example, they are doing directed evolution of thermal stability, so they are comparing activity with and without a heat challenge. You then write out some equations. You have one equation for each clone that you assay. The measured fitness for each clone is y. Each x1a-like value is either 1 or 0 for the presence of each possible base substitution. The C1a-like values are the fitness contributions associated with each mutation. These are unknowns that we are trying to measure or solve for. If you assume that the contribution of most mutations is additive, then you can describe the fitness observed for each variant as a linear combination of the other mutations. In practice, this additivity is usually true with the exception of mutations in the active site of the protein. After solving the equations for each C value, you do additional design of experiment calculations to choose those mutation combinations that will be most informative to test during the next round of mutagenesis. In addition to a mutagenesis strategy, your directed evolution experiment requires a screening method. In a company that specializes in directed evolution, they may have high-throughput mass spec instruments that can do LC-MS assays at more reasonable throughput, and thus many chemical transformations can be observed. Effectively, this is doing less precise but faster product studies on each mutant. For the less expensive experiments often done in academia and small companies, plate-based methods using colorimetric or fluorometric assays are the norm. For example, you might fuse GFP to your protein to link the amount of your protein to a detectable signal. You might also use cell-permeable substrates, as Arnold did with the paranitrobenzyl esterase. Thus, these are analogous to the whole cell assays we previously discussed. Typically, you will want to grow the colonies in 384-well plates to normalize the amount of cells and then perform these assays on those more consistent cultures. This can be done somewhat easily at the 10,000 sample scale. Basically, any quantitative assay you can do on a single strain could be considered for use in directed evolution. The only concern is how many of those assays your labor force and materials cost enable you to do. There are companies that optimize protein expression by running each variant on a gel. Though that sounds low throughput, if the diversity you are able to pay for is lower than the diversity required to obtain the data you need, that assay will do. There are all sorts of things you can use directed evolution to improve about a protein. You can improve its catalytic efficiency sometimes, but more typically you improve expression or solubility of your protein. You can also improve the catalytic parameters, usually Km, for a non-native substrate. You can also improve the thermostability, buffer stability, and solvent tolerance of the protein. In a nutshell, any application in which one or two point mutations give rise to a detectable change in activity and a selection or screen can identify those changes can be accessed with directed evolution.